Good day. I'm Janet Vaughn, Teva Pharmaceuticals, and I'll be presenting an industry perspective on generic product development challenges and research priorities for the next 5 years. The intricacies associated with complex generics present significant challenges in the development of these products and achieving regulatory market approval. There are opportunities for research leading to better understanding and acceptance of the science necessary to bring complex generics to market expeditiously. This presentation highlights challenges in the development of various types of complex generic products as well as recommendations for resolution. Impurities in generic synthetic peptides, which refer to a recombinant RLD, are differentially expressed to the reference product. From this analysis, new impurities or impurities that are also present in the reference product but at a higher concentration are handled on a case-by-case basis using a totality of evidence approach, including in vitro studies to evaluate immunogenicity risk. But there is minimal guidance available for in vitro immunogenicity study design expectations, and the specific details are often communicated in complete response letters. So, our recommendation is from a public domain standpoint, if FDA could develop multiple publicly available validated methods for innate immune assays and DCT cell assays that the agency finds acceptable, this would likely reduce the number of deficiencies in cereals. Additional specificity and guidance for study design considerations would enable sponsors to conduct studies in line with FDA's expectations. With regards to iron colloids, comprehensive side-by-side characterization studies need to be performed on the drug product, the iron core, carbohydrate, and label iron under physiologically relevant conditions. Differences between the products must be thoroughly investigated and appropriately justified. There are, however, no publicly available analytical methods for in vitro characterization, none that has been established by FDA. So deficiencies in cereals often relate to the way in which the products are characterized and insufficiency of characterization. Only during the review procedure does FDA request industry to follow specific methodology. The requirements evidenced in various PSGs are continually changing, and the in vitro parameters that impact in vivo performance are debatable. So, our recommendation is that further research is done to develop appropriate methodologies with sufficient level of detail for in vitro characterization, and also to develop guidance to communicate FDA study design preferences specific to iron colloid products and facilitate further evaluations of in vitro parameters that are clinically relevant. For long-acting injectables, FDA has indicated an interest in seeing modeling and analysis plans in terms of model-based approaches for bioequivalence assessment, if proposed, as part of pre-ANDA submissions to obtain concurrence prior to execution. However, it has been highlighted that information requests are common for these types of pre-ANDA meetings, and this takes away time from the assessment clock. Model integrated evidence can have a meaningful impact on reducing study durations, but the specific expectations are unknown. So we recommend that it would be beneficial to both industry and FDA if there was a mutual understanding of the information to be submitted in pre-ANDA meetings, to make the most of the meetings and to set a strong foundation for data to be generated in support of an ANDA. So it would be helpful if guidance is developed that is specific to model integrated evidence with suggested approaches, designs, and templates for submitting the information in an ANDA. For drug device combinations, there is no available guidance that represents FDA's current thinking on comparative use human factor studies or even how to properly calculate a non inferiority margin to employ in these studies. So FDA's current expectations for comparative use human factor studies needs to be publicly communicated. So we recommend that regulatory science and research regarding acceptable study designs and NI margins that can be employed in these comparative human factor studies. And this could include workshops, training, and focus groups that would be beneficial to both FDA and industry. Research in this area could facilitate a common ground whereby other differences between test and R&D combination products could be effectively managed and not preclude approvability via the 505J pathway. Challenge for respiratory drugs is the move to environmentally friendly propellants. Under the Kigali Amendment to the Montreal Protocol, production and consumption of HFCs will be cut by more than 80% over the next 30 years. But there still is no clear guidance from FDA as to what is needed for generic drugs in the event the RD switches to a greener propellant. Industry needs clear guidance from the agency. Will generic manufacturers be required to switch to propellants in line with the RD? If required, what studies will be necessary? Will in vitro equivalency be sufficient to support a switch? Again, for respiratory drugs, are there opportunities for utilizing CE waivers? There can be reduced in vivo data requirements following establishment of improved modeling, but there is no clear guidance provided in product-specific guidances for CE waivers. So the recommendation is to develop general expectations for CE waivers, product and data requirements, and to provide adequate in silico modeling parameters needed to be outlined, minimum requirements concerning alternative in vitro methods. This would allow product development meetings to be a discussion of generated data rather than theoretical proposals. With regards to transdermal systems, manufacturers often make changes in adhesive composition, the type, the grade, etc., while maintaining the same acceptance criteria. However, any change to an adhesive is considered major, thus potentially impacting all stages of lifecycle management. So we would like to see acceptable limits developed for changes in adhesives and the data requirements to support the change. Another challenge is with changes in drug release specifications, these changes may require repeat of PK studies. However, for a large number of products, IVPT is a valid tool to establish bioequivalence. So can the IVPT be used for justifying any changes to the in vitro drug release specification? So what we would recommend is the development of a mechanism to compare or correlate IVPT and in vitro drug release. For semisolids, variability in RLD in vitro BE performance makes it difficult for RLD lot selection for IVPT studies. So the suggestion would be to establish criteria for selection of the RLD lot and some guidance on how to account for the observed variability in the final study. Another challenge we see for semisolids is a lack of procedures for addressing aberrant non-robust data points for IVPT studies. So it would be beneficial to establish a methodology to address these aberrant data points during IVPT studies. Now, preservatives have a tendency to adsorb to the applicator, and the level of preservative decreases over time. Depending on the age of the R&D lot, it is difficult to match the preservative content of the test product against the R&D for Q1, Q2 sameness. So our recommendation is to consider utilizing preservative efficacy studies to support Q1, Q2 sameness. Other general challenges are, for instance, a lack of global harmonization. This makes it difficult to develop a product for the global market. Product-specific guidances. There's still a lack of product-specific guidances for certain drug products, and often the PSGs are unclear. So further refinement of PSGs such that there is clear understanding regarding FDA's requirements for approval would be beneficial. GDUFA 3 promises to improve issues associated with PSGs, but we do have some work to do. The lack of SUPAC guidances for various complex dosage forms makes it difficult for industry. So establishing SUPAC guidances for all complex dosage forms should be worked on, should be made a priority. This will not only assist the industry, but will also reduce the number of requests to FDA for guidance. So in conclusion, while much has been done, some challenges remain. There are opportunities for further studies to remove or alleviate these challenges. Industry looks forward to further discussion and to GDUFA 3 implementation. I would like to acknowledge my colleagues at Teva, Brandon Woods, Alan Thompson, Virana Lola, as well as to the Teva Research and Development team. Thank you for your time.